What we're talking about is something that we believe may be as ancient as life on the planet itself, if not life even on other planets themselves, right up to the very latest technology can offer us. So if you have a cell phone there and you have a digital capture on it, you should be able to pick this up. Those of you who have one and want to try it, thank you, Lyndall, nicely done. You should be able to pick this up, and if you've got the right software, it should deliver to your iPhone screen a copy of the publication itself. So we'll put it up again at the end if you want to try it. I'll leave it there for a second for those who want to. Yes, Richard?
If we do that, can we disappear?
Yes, but you'll have to do the exam afterwards just the same. All right, so that's there, and we thought that was kind of cute as well. So the talk's broken down into a series of steps. As I said, we'll talk about roughly why are we doing this, because it's interesting, and what do we want to achieve. We want to see if this model really has some sort of universal applicability, but also how that might then be used. I'll talk about the basic idea behind the model that we're using. Ross will talk in detail about the modelling procedures that are being used to fit 230 quite disparate data sets to one model with 8 or 9 parameters, depending on how you look at it, and what that process tells you at the end of it. So again, Ross will go through the results, and then we'll talk a little bit, speculate a little bit about what it all might mean. If I can get the next slide to advance, there we go. So anyone who does work with living systems that aren't homeotherms will know that as the temperature goes up, they tend to do things faster, whether that's growing or running or slithering or whatever it is they do. And there's a general temperature response that most organisms display. That is, as the temperature goes up, whatever they do faster. We're talking about growth most particularly. The data we've used in this process is growth rate data. So as the temperature goes up, the rate of growth goes up faster and faster and faster. It accelerates ever so slightly until it gets to a maximum level, if you like, at which the rate is as fast as it's going to get. And then typically, as the temperature gets higher again, that rate declines. So it gets slower and slower and slower until eventually it stops growing. Some people call it Tmax, the maximum temperature for growth. The one that this extrapolates down to Tmin, the minimum temperature for growth, we think. And this one, where growth rate is fastest, Topt, optimum temperature for growth, which in fact turns out to be a bit of a misnomer, as we'll explain in a minute. So what we're trying to do with this approach is say just how common is that basic pattern of organism or biological response to temperature changes. And the idea was to get as many data sets as we could, because in a previous paper we'd speculated that this response really was quite universal. Now, one of the challenges we have is that there are so many different ways and units of measuring rate that what we've actually done is normalise the data to say what we're going to do is fit the model to data that's been normalised so that every rate that we observe is relative to the maximum rate of growth of that organism. So we've got basically a whole set of data, 230 data sets, all of which scale from 0 to 1 and back down again depending on temperature. Again, that makes it a little bit easier to do, makes the modelling process a little bit more straightforward and achievable. And why do we do it? Well, our interest originally, so you saw right back on the first slide, I should probably, if I knew how to go back, I don't know how to go back. June and Tom McMeekin and David Ratkowski and I have been working on it for a long time. Ross came to join the group and applied his particular modelling skills, and then John Bowman's become involved as well, applying his understanding of biology and microbial physiology. But the original inception was, how do microorganisms respond to temperature changes? Because that's really relevant to the spoilage of food. The faster they grow, the sooner it goes off. Or to the safety of food. The faster the microbes grow, the bigger the chance that you'll get sick if you happen to eat some that are contaminated with pathogens. But now that we've been able to extend this to a whole range of other organisms, and it's not just microbes, it actually extends to insects and other kinds of multicellular organisms as well, and plants. And so there's a lot of interest in, well, what happens if the temperature really does go up a few degrees? And the importance of this might be, well, if we do have this universal relationship, and we know pretty much where along the temperature scale a particular organism is adapted to, if the temperature on average goes up by 2 or 3 degrees, probably doesn't matter too much if you're in this range for that particular organism. It's just going to go faster and be happier. But if you're up about here and you go over 2 or 3 degrees, we might find that there are some negative consequences for the activity of that organism in its ecosystem. And so there is a fair bit of interest around the effects of temperature on biological processes, because we expect that temperatures around the planet are going to change. This could be a useful thing to do. So we got data from lots of places. We had a lot of our own in-house for microbes that grow in foods and are relevant, others that we'd collected, others that others had collected, and then Tom and others actively searched and scoured the literature, Ross decoded it and put it into data that could be used as well. So there's an awful lot of information that has been collected for 230 different strains of living things, not just microbes, but insects and mites. Do we have plant tissues in there as well? One plant. And I'm pretty sure that if we got lots of additional plant information, it would do exactly the same sorts of things. So again, that's the basis. There's an awful lot of data to be fitting to a model at one time. As I mentioned, we use the rescaled rate, so we have a relative rate for every single of those 230 data sets. As I said, there's different ways of measuring rate, but that normalisation process gets around it, so really all of these data sets are directly comparable, and effectively what we're doing is saying how reproducible is the shape and the proportions of that growth versus temperature curve that I showed you a moment ago. For reasons to do with statistics, that is, some data at different temperatures will have more variability in them. We normalise that variance by taking a square root transformation, largely through the work of David Ratkowski, who discovered that was a really relevant thing to do for microbial growth rate data. We might possibly have also done it just by taking a natural log transformation as well, which seems to achieve the same sort of function. But historically we've used square root transformations because we know they work. So the basic idea of this model is that it's trying to explain the shape from basic ideas in thermodynamics and chemistry and enzyme catalysis. And what we're saying is that, yes, temperature governs the rate of any sort of chemical reaction. Equally, it will govern the rate of a biochemical reaction, something that's being catalysed by a biological catalyst. And so what we're saying is all of the systems in life that we see are being driven by enzymes as biological catalysts. Those catalysts actually make life reactions possible. Without them, they'd probably go too slow for anything much to happen at all. So in a way, enzymes are nature's dating service for molecules. What an enzyme does is to bring down the activation energy of a reaction to make it go at an appreciable rate so that life processes can happen. Now, if you go right back to your basic chemistry, maybe in grade 12 or even first year uni, you probably will have encountered a thing called an Arrhenius plot, which is a graph of the response of the logarithm of rate of reaction versus the inverse of temperature, where temperature is measured on the absolute scale. I won't try and explain why we do that, but it's been used for 130, 140 years in chemistry with fairly good success. And it basically says that if you do this transformation, if you plot the log of the rate of the reaction against the reciprocal of the absolute temperature, you get a straight line response. And that straight line gives you a measure of what's called the activation energy, which is the energy required of the reactants to come together and actually complete that reaction process. So you've got molecules bombing around in solution. As long as they're jiggling around hard enough, occasionally they hit either in the right orientation or with sufficient energy that they react, and that's basically what we're measuring here. What's the probability? How does the rate change as a function of temperature? The hotter they are, the more energy they've got, the more likely that the reaction will proceed. Now, what an enzyme does, a biological catalyst, is to increase the likelihood that those reactants will meet together in either the right orientation or the right space. Enzymes reduce the activation energy. They reduce the amount of energy required by each of those reactants for that reactant to proceed with a reasonable amount of confidence. So enzymes are critical to a lot of biochemical processes. But what you find is that if you do plot the rate of reaction of an enzyme catalysed process, it doesn't follow that straight line relationship that the chemists expect. Chemists expect this to continue for hundreds of degrees without too much deviation. It's actually got a slight curvature in it, but we won't go into that. If you look at biological reactions, they follow that Arrhenius kind of prediction for a while, but as the temperature gets lower, they deviate quite markedly, and as the temperature gets higher, they deviate quite markedly as well. And the explanation for that, that's probably been around for 60 or 70 years, is, well, the enzyme, the thing that actually reduces the activation energy, is itself a complex molecule. And as it is affected by temperature, its shape changes. And we all know, I think, that the function of an enzyme is very much related to its three-dimensional structure. It has to bring those two reactant molecules together in just the right orientation, and perhaps even bring them even closer than that, and that's how they work. But if you distort the shape of the enzyme, it doesn't do that job so well anymore. So as the temperature goes down and the enzyme starts to freeze up, it doesn't bring the reactants together in the right orientation, and if it gets too excited because it's too hot, same problem. So low temperature, there's a deviation in the conformation of the enzyme. It's called cold denaturation. At high temperatures, you get the same thing. If you go too high, you get irreversible denaturation, which is when your egg fries. But you can have the protein move into that higher temperature region, lose its conformation, lose its functionality, but up to a point it can still come back again. So the basic idea is whatever that reaction is, the enzyme's catalysing it, that's what it will do as long as the enzyme stays in the active conformation. The enzyme can't because it's a biological molecule and we get this pattern. This is what we're trying to model. Okay, explanations for that are that, well, that's what I just said. There's reduced growth rate and efficiency because the enzymes aren't working properly. The same up here at high temperature. In this region where it behaves pretty much like a simple chemical reaction, we call that the normal physiological range. The range where that organism or that reaction process is quite happy to continue and behave as if it were simply an ordinary chemical reaction. Now, it's a lot easier to look at the data this way, and again, this is just showing that typical pattern of response that I produced in the second or third slide. But what we also know is that different organisms are adapted to different ranges of temperature. They all have the same shape, but some prefer to grow at cold temperatures, others can tolerate quite high temperatures. But the same pattern pretty much holds, we think. And so really what we'll demonstrate is we think it even more strongly now. So one of the things we have is how does all of that fit together with this universal model, and does it really work? But to go and take it another way, if I translate that same idea about enzyme catalysis into this form of just growth rate versus temperature, this is what the AINS would predict that chemical reaction would do, that master enzyme catalysed reaction. But we also know that the enzyme that's catalysing it has a limited range of stability, which looks something like this. So what we're saying is, in this range, the probability of the enzyme being in the right shape is about 100%. But as the temperature gets too cold, that probability drops off so that there's actually a reaction between the inactive and the active form, and it's a function of temperature. As the temperature rises, the probability of the enzyme getting into the active form goes up until it's in this range, but at some point the probability comes down again. So you've got an enzyme that's pretty active here, but as the temperature goes down or up, it loses activity, or catalytic efficiency, I guess. So if you superimpose those two graphs together, this is what you get. So if I take this information and correct the predicted rate for the fact that actually there's a whole lot less enzyme doing the job now, you end up with a curve that looks just like this, which probably looks fairly familiar. The other thing to note is, right at the outset, I said up here we call this T-opt, and I said it was probably a bit of a misnomer. At T-opt, this cell, this organism, is going about as fast as it can. It might not be doing it terribly efficiently. It might be like a car that's racing and revving very hard. It's burning up more fuel. It's going faster, but it's not going ultimately as efficiently. If the car were being nursed, we'd be down in this T-mes range. So T-mes is interpreted as the middle of the normal physiological range. It's that point where we would expect that cell to be growing in the happiest possible environment it can, with most of its systems functioning as efficiently as they could. And that's just reinforcing it again to show this is pretty much the shape of the curve that we showed, the one that we observe in all the data sets that we've got. And the reason for that, we think, is that explanation that I just said. There's an enzyme catalysed reaction. The enzyme itself is affected by temperature, and the consequences are this is the overall shape of the response that you get. Enzymes warming up, enzymes optimally active, enzymes getting a bit hot and bothered, comes down again. So the consequences are that's the shape of the curve. So back to where we were, looking at it from a more traditional AINS perspective, because that's the basis of the theory that we've used to develop the model. And when I say we've used that theory to develop the model, it's certainly we're not the only group to have done that. These ideas have been around, as I said, for 60 or 70 years. What's a little bit different is we applied some new understanding of protein thermodynamics to the way that we structure our model as well, which in principle means, I think I said all of that, yep, which in principle means that if we could measure these parameters here, there's quite a few of them independently, and these ones, these are properties of the enzyme's thermodynamics as it moves between the active and the inactive state and the reaction itself. And if you could measure each of those properties independently, which in theory you could, we could then predict the overall rate of growth of the organism. So this is the structure of the model. It's based on an AINS equation or an AINS prediction of reaction rate corrected for the fact that the protein catalyst itself is subject to the effect of temperatures. So if you like, this part of the model is a simple Arrhenius model. It says in the absence of denaturation of the catalyst, that's what we'd expect to happen. The denominator is a rather complex equation that says what's the probability that the enzyme will be in the active conformation as a function of the temperature itself. And that's based on some work that was done maybe 15 or 20 years ago, looking at how proteins change as they become denatured. So the heat capacity changes when the protein folds up, is a measurable change in its capacity to hold temperature, if you like, or to hold energy. And these other parameters relate to particular properties of proteins as they get to a certain temperature. And they are thought to be common to all protein-based systems. So any protein molecule should have the same values for those 4 parameters. This one is a function of the enzyme itself, so it's unique to that particular enzyme. So again, this is really just a mathematical way of saying what I showed you in the graphs that I showed you before. But what we're doing is not measuring the thermodynamic properties. What we're doing is taking growth rate data and temperature data and saying, can we, from that data alone, regenerate these values? And that's what this modelling exercise has been about. And the reason that you would do that is not because you're particularly interested in them, but if you can show that they are in fact constants and that they do behave in the way that the theory predicts, it lends some credibility to the model itself. It actually says what we get out of this process, which doesn't involve thermodynamics, it just involves growth rates, produces values for those predicted thermodynamic properties that kind of make sense. So as I said, there are 8 of them. Sorry, I forgot to mention one. This one relates to the number of amino acids in the protein, and that's an interesting one that you'll talk about in some more detail because it's got some interesting speculative potential. So as I said, 4 of those we call strain parameters. These are things that are unique to the living system itself that we're talking about. That's the activation energy of the reaction that's being catalysed, the number of amino acids in the molecule, the change in heat capacity, which is a function of the structure of the protein itself. And this is a constant that sort of is a bit of an index of what's the likelihood of these molecules colliding in the right sort of orientation and at the right temperature that they finally react. And we kind of eliminate that a little bit by normalising the data. And as I said, also, there are these 4 universal parameters that we believe should be constant for any given protein molecule. So, to reiterate, because I'm about to finish, what we're saying is this is the model, this is the theory it comes from, and this is the process that we're going to do. And so the modelling process itself has been to say, does the theory behind this model hold up when you throw an awful lot of data at it? And I think at that point, just reiterating what I just said, I can throw over to Ross, except to say, as I said earlier, we're not the only people to come up with this idea. All we've done is brought together some different publications that enable each of those parameters to, in theory at least, be independently evaluated. And with that, I'll hand over to Ross. Thank you.
Does mic work?
Sorry?
The mic works.
I'm not so sure. Should do, but if you stand close to it.
Yeah. Is this mic? It does work. Right. Right. Before I describe what's on the screen, I need to backtrack a little bit. In 2005, there was a publication, Ratkowski et al., which used the same fundamental equation as is on the screen. And it used 35 bacterial data sets. Each of those data sets was analysed individually. So you've got 35 strains and you've got 35 different analyses. The model was as good as could be done at the time, but it wasn't complete. They couldn't estimate all of the parameters simultaneously in each of those 35 different analyses. Subsequent to that, I became involved and I was asked to see what I could do, and I put it onto a Bayesian hierarchical framework. That meant we could integrate many more data sets simultaneously and analyze all the parameters simultaneously. The number of data sets that came out in that publication, which was in 2012, was about 90. Now, the point about that is that all of those parameters in all of those strains were estimated simultaneously. I'll just go back a slide. Other way. These ones here were called the strain parameters. So they could vary per strain. So if you've got 35 or 90 or however many data sets or species or strains or whatever you want to call them, each of them had their own unique value of these 4 parameters. But they all share the same values of the universal parameters, which is why we're calling them universal. Now that's something you couldn't do without using a Bayesian hierarchical framework, so that's an advance in itself. Now, subsequent to the 2012 paper, where we had included bacteria, archaea, and eukarya, which are the 3 domains of life, that means we're covering all of life. All of life is in one of those 3 domains, but they were all unicellular organisms. And the eukarya, we cheated a bit, we just included fungi. But that was very encouraging, so after that we went to the next step, and I spent quite a long time dragging up data on whatever I could find. And that was mainly insects, because the implication is that if it works for such a variety of unicellular organisms, it might possibly work for multicellular organisms. And insects are a convenient organism to pick because entomologists go and measure them because they're of economic importance, they're of agricultural, medical, veterinary importance. And so a lot of veterinary papers will list the growth rate using intrinsic growth rate rather than specific growth rate, but they're logically the same. So having done that exercise, we ended up with 230 data sets. Now, another aspect of borrowing of Bayesian hierarchical modeling is that some of these data sets don't have much data in them. Some have lots of data in them. You might have a bacterial data set where it's easier to measure rates, and so you have lots of data points. But then you have an insect data set where it's very hard to do the same thing, and so you may only have 5 or 6 data points. The advantage of using a Bayesian hierarchical framework is that you get information flow from the data sets with lots of data in them to the ones which have fewer data. Now that's called borrowing strength. It means that you can actually fit curves with a complicated expression like that to a data set which has only got half a dozen data points on it. But collectively, these 230 data sets contribute information that they exchange. I'm talking metaphorically. They exchange information between themselves, and they also collectively contribute information to estimate the universal parameters, which are very hard to estimate. Unless we had 230 parameters, we could do it with 90, but with 230 we can do a much better job. They're so hard to estimate that we need a lot of data to do it. Now, we did use rescale data, and Tom alluded to that. There's several other reasons for why we did that. One is that some of these original data sets we're using units of varying types. So if we rescale to 0 to 100%, that gets around that problem. But a more subtle problem is that my original intention following the 2012 paper was to try and emulate what's done in the metabolic theory of ecology, which I'm not a fan of, but I thought I'd steal some ideas out of there and scale the rate to as a power of mass of the organism. Now, according to the metabolic theory of ecology, metabolic rates or growth rates and all sorts of things are supposed to scale with mass to a certain power. This idea originally stems from Kleber in the 1930s. And if you go back and look at the Kleber paper, he doesn't say that the metabolic rate scales with the 3/4 power of mass. He doesn't actually say that. He scales, according to him, it scales at about 0.71 or something like that. He rounded it to 3/4 for computational convenience, which makes sense if you don't have computers and you just have slide rules and whatnot. It's easier to use 3/4. But since that time, people have often assumed that it's a physical constant. But it was actually just a rounding convenience. Another reason why 3/4 is problematic is that it's a bit controversial. If people go and measure it using ecosystems and individual populations, it doesn't come out as 3/4. It comes out as all sorts of values between 0.6 and 1. So why use 3/4? We get around the issue by rescaling the data, but it is an important point which we'll have to come back to in the future. These are the categories of the data sets. Also following the 2012 paper, we initially planned to do what we did in the 2012 paper and just divide the group species up, strains up into the 3 domains of life. which is bacteria, archaea, eukarya. But it occurred to me that it makes more sense to assign them into groups which are based on their ecological adaptation. So if they're adapted to a high temperature environment, let's put them into a high temperature group, and vice versa for cyclophiles, which are a low temperature group. So initially I thought, let's just use the standard categories: cyclophiles, mesophiles, thermophiles, and hyperthermophiles. And the standard cut points, which we chose, they are somewhat arbitrary, but maybe they're not. Maybe they have some biological meaning, of 17, 45, and 80. And we fitted a model using those breakdowns, and it worked much better, which means that we were better accounting for the variation in the data, except for the fungus. Now, the fungi refuse to cooperate. They're very annoying. As a result, we've got 5 categories. We did actually go through a model selection process where we had a model with no categories, just one domain. In other words, the 3 taxonomic categories, one with 4 thermal groups, one with the 5, and there's a 5th combination. I can't remember what the last one was. The best one was this breakdown. The way we assign these species to the strains is by looking at the temperature at which they grow fastest from the raw data. You plug them into the right thermal group. We fit the curve. Once you've got the curve, you can recalculate what the theoretical T-opt is, and then you can reshuffle them around a bit to get an improved categorization, which makes a small difference, but not a big difference, but it does improve the model. We're still covering all of the 3 domains of life. They're still in there, but now we've got insects and mites. That's very ambitious. At the time, it seemed very ambitious. We have 1 colembla, because I've never found any other data. I've only got 1 data set for colembla. The data set's very diverse. It contains acidic organisms that like acid conditions, salty conditions, and alkali salty conditions. It also spans a huge temperature range, 124 degrees. So that's very big. But that goes from -2 degrees, below which we get, it's difficult to measure rates for various reasons. But we do go up to the maximum temperature of 122 degrees. For Methanopyrus candeleri, it actually grows up to that temperature, which is well above boiling point. And you may wonder why, but this is an organism that grows at the bottom of the ocean under high pressure, where it's possible to grow at above boiling point. It doesn't like growing below 80 degrees. It's uncomfortable. It thinks that's a bit chilly. This is the equation again. Need to put some more structure into it. These 5 thermal groups were assigned their own set of parameters, just like the strains are assigned their own parameters, the same parameters, in fact. It's convenient to think of the thermal groups now as representing the average behavior of the strains they contain. So we now have 3 sets of parameters. We've got strain parameters, 1 set per strain, the thermal group parameters, so there's 1 set per thermal group. They're the same parameters, as it turns out, and the universal parameters which apply to everything. This is, and so yet some more definitions. Here we've got just a standard data set and a fitted curve through it. This is the T-opt, which is the temperature at maximum rate of growth. This is T-mes, this is the protein denaturation curve, and this is T-mes, and superimposed there. The curve's about halfway up. We also define, for convenience, the 50% denaturation temperatures, which are TL and TU. As you notice, this one is down in the tail, and that one's close to the peak. Now, I need to explain a little bit about how I fit the model. This is the Bayesian model. The most commonly used technique in Bayesian modeling is Markov chain Monte Carlo, or MCMC. It's a simple technique. It's very easy to describe. We start, say, with 2 parameters, and supposing we're modeling these contours, supposing they represent a probability distribution of x and y. What you do is you start out with a guess, as you do in nonlinear modeling. You have to start in a reasonable guess, and you just try and change one of these parameters. You fix x, and you take a guess for what a new value of y is. You calculate its probability in the context of the model, and you essentially flip a coin and see if you can move. And if it does move, you keep that value, and then you try and change x. And then you start with it, you go back to y, and you go back to x. It's a very simple technique, and it works most of the time. It doesn't work with this model. We've got over 1,000 parameters because each of those strains has 4 parameters. Essentially, each of those strains embeds that original equation that I mentioned. Each of them has their own little equation. So there's 1,000 parameters, and they're highly correlated, and especially the universal parameters. So we can't use simple MCMC, though it's still in there. And we have to use more complicated methods, such as HARIO updates, adaptive directional sampling, and stepping stones, which would take too long to describe. Now, this is a Bayesian model, which is different to frequentist or non-Bayesian models you may be familiar with. The main distinction is that parameters have prior distributions, statistical distributions. That this prior distribution represents what you know about something before you look at the data. In non-Bayesian frequentist statistics, you're supposed to be objective and analyze data by themselves, but in Bayesian statistics, it's different. You say, well, we do know something about those parameters. If you've never seen a cow, you could probably, but you've heard of them, you could probably guess that they weigh less than 10 tons and they weigh more than 1 gram. So you do know something about almost anything. That's the same principle we use here. We assign non-informative prior distributions to the parameters. So we're not telling the model what to say. We let the data speak for themselves because these are non-informative prior distributions. Each of these parameters is assigned a uniform distribution. And this one is temperature. We're saying it's got to equal its probable values can lie between 320 and 420 degrees, and any value in between with equal probability. So anything in that range is fine. We're not prejudiced as to which value. The same with each of these other distributions. They're very much wider than they need to be. They've actually got wider since we started. Originally, they came from literature guesses. So this one, for example, n, 1 to 20,000, which encompasses, it says far greater than it needs to be. All we're saying is that it shouldn't look outside that range. So we're not telling the model what parameter values to use, but we're just giving it a hint as that it should be within certain extreme limits. Results. These are the universal parameters. And I'll start with the least happy one first. The first one is enthalpy change, delta H star. If you measure globular proteins, this value would be about 6,500. Comes out as 4874, which is lower than it should be. But we don't know what this reaction system, if it is a single reaction system, we don't know what the nature of that system is. It may well be 4874. So it's not as if it's 14,000. It's a good value. What's really nice is that the next one down, entropy change, is 17. Experimental values for globular proteins is 18. Now, we didn't tell it what values to come up with, and it comes up with 17 instead of 18. I think that's quite nice. The temperatures, the experimental values for globular proteins are around 383, I think, or 393 and 385. Close enough. I mean, they're plausible values, given that we don't know this is a globular protein. It's not as if they're wildly different. They're plausible. And in fact, it's worth noting that these, you can think of these as confidence intervals. They're actually credible intervals. That's only 1 degree wide. So the model's insisting on these values. It's insisting that this parameter has that value. So it's, if anything, it's probably indicative, or might be help in indicating what the nature of the system actually is. These are some examples of the fits. It's on the square root scale, but you'll notice this is for the hottest organism known. It grows at up to 200, whatever it's at, 120 degrees. Good fit. And that's, as you see, it doesn't like going to cold temperatures below 80. Here's a psychrophile. It also fits very well. It likes cold temperatures. And this is a fungus, which fits quite nicely. These are some more examples. Now, if you did get the barcode to work on your phone, you can look up on your, you don't have to wait to get back to your desk. You can look them up straight away. We've got a lot more graphs in the paper. Here are the archaea, and you can see they're lovely fits. They really are nice. These are the bacteria. They're very nice, some of them. The first are the fungi, which are very strange. But these are the insects and the mites, which are multicellular. So even though they're in the model, they're still fitting. Now, if you look at it by thermal group, psychrophile, as you'd expect, is at the lower temperatures, then going up through fungi, mesophile, thermophile, and then over here are the hyperthermophiles. So it gives you an idea of the distribution of these things. But what's interesting now is that we have predictions about what the nature of the system should be. This is the protein denaturation curve for the 4 thermal groups. Psychrophile, as you'd expect, is down at the lower temperature range, but you notice it doesn't go to 100%, which we speculate might mean that perhaps psychrophile proteins aren't that stable. Maybe they're slightly inherently unstable, so they never get to 100% fully denatured, or maybe they're not being replaced enough. Mesophiles and fungi also don't quite get there, but they are nevertheless sort of centered on each other. Then you go into the thermophile and hyperthermophile groups, and they become very flat-topped. Now that's something we didn't expect. They also join up on the bottom, the lower temperature range, so they become more and more stretched as you go along. So the suggestion is that proteins for thermophiles and hyperthermophiles are becoming more and more stable over a wider and wider range. If you look at the quantities I described earlier, essentially we're looking at the same information again, except more quantitatively. Tmes is the temperature of maximum or minimum denaturation. T-opt is the maximum rate of growth, and TL and TU are the 50% denaturation temperatures. You'll notice that T-opt and TU minus T-opt is almost a constant. It doesn't change much. What that means is that as the temperature goes up, the maximum rate of growth is pushing right up against the maximum, the TU. It's as if it's on the edge of catastrophe. It can't go any faster. It's really pushing that boundary. And it's leaving way behind the TL, which becomes increasingly distant from it. The Tmes is central to it, to the whole thing. So as enzymes adapt to higher and higher temperatures, their optimal thermal activity is very close to what the protein can actually do, assuming that there is just this one protein. If you look at the prior estimates for the thermal group parameters, then we find that, we'll start with the strangest one first. This one is the delta hedge, I say delta hedge dagger, enthalpy of activation. You notice there's a trend going, sort of a trend going up to 48 to 96. Definite trends here going up and down. The enthalpy of activation sort of trends with temperature, except for the fungi, which are again are a bit of an annoying difference. Other studies, however, when they've actually measured this quantity using other modelling approaches, also find that this quantity is rather erratic. They don't come up with one value. It's all over the place, except for the metabolic theory of ecology, which assumes that it's a constant, which is not true. If you look at that particular parameter and you graph it out against T-opt, here's your hyperthermophiles, thermophiles, psychrophiles. You can imagine a trend there, all from fungi, which are very low. What's odd is the mesophiles and the thermophiles are constant or similar. Whether this persists when we have an even larger data set would be interesting to see. The value for the fungi are curious. Maybe that's a speculation, maybe that's something to do with prion-like proteins. But what's certainly not the case is, like the metabolic theory of ecology usually indicates, it's not a constant. The delta CP is a very strong correlation with T-opt, but it's not a perfectly straight line. It's got this curious curvature about it. But it goes on very nicely from psychrophiles, mesophiles, thermophiles, hyperthermophiles, and the fungi are where they, for once, where they should be. This makes sense in that delta Cp, heat capacity change, should be correlating with the stability of the proteins. So my explanation for this is that as the T-opt is increasing, as the temperature is increasing, the molecules are banging together faster and faster and harder and harder. proteins have to be more rigid, more stable in order to do what they do. So a hyperthermophile, and thermophile needs more rigid protein, so it needs a delta CP which is higher. Psychrophiles have to sacrifice that for greater activity, so they can afford a lower delta CP. Experimental work does indicate delta CP increases is higher in thermophilic proteins versus other lesser temperature proteins. So this conforms with what we expect from experimental work, even though, I'll remind you, we're dealing with the biological growth rates of insects and bacteria. But we're getting out relationships which come out of protein chemistry. I think that's remarkable. Here's protein length. Experimental work indicates it decreases it's shorter, they are shorter, in thermophilic organisms versus psychrophilic, etc. It's the same trend we actually predict from the model. And this, in my layman's interpretation, would mean that as the temperature goes up, a larger molecule is harder to keep stable. So it makes sense to have a smaller molecule. The total heat capacity is what's usually reported in the protein chemistry literature. So I've calculated that, and that also goes down as it's supposed to according to the biochemists. Hydrophobicity, the NCH, is, once again, according to the biochemists, it's supposed to go up, and it's predicted to do so. So we're doing well. So the interpretation of this is that if you've got a low temperature environment and a psychrophile in that environment, you don't want the delta H dagger to be too high. You want the molecules have got to jump over an energy barrier to react. So you don't want that barrier to be too high. But you can afford to have a lower delta CP. As the temperature increases, the cell wants to keep the reaction rates in balance, so it needs a higher delta H dagger to essentially dampen down these reactions. So it needs a higher delta H and a higher delta CP. The N also makes sense in terms of the size of the proteins. But I'll remind you, we are getting results about organism denaturation curves, but we're not using protein data at all. We're using growth data of insects and plants, or one plant. Now, hyperthermophilic proteins, we predicted with a denaturation curve, should have a flat top. Do they? That's the obvious question to ask. Well, there are certainly proteins that do. Kinases from Thermophilus is almost flat over a 60-degree range compared to the same sorts of proteins from a mesophile where they're not, they're peaked. Similarly, cut-A1 from a hyperthermophile is more stable over a wider range than its lesser temperature equivalents. This is a particularly nice one. The serum albumin in this particular study from Cordova 2008 is extracted from sheep, but it's stable at 150 degrees. Now, why a sheep needs to have anything stable at 150 degrees, I don't know. But it's possible, as some people have suggested, that life originated from a thermophilic organism. Now, if all life started from a thermophilic origin, and we've since adapted to lower temperatures, we may still be using some of these proteins which have been conserved over that period of time. So that suggests the possibility there's other proteins of a similar nature which are still around in lower temperature organisms. Now, as I've kept saying, we have insects in this data set. So we've got one model which covers unicellular and multicellular organisms. The curious thing is that multicellular, you would think, multicellular reproduction is a bit more complicated than in a unicellular case, but I suppose it depends on your point of view. But there are several mechanisms which may come into play or be related. One of them is the observation that development stages in insects, such as the embryological stages and the larval stages, the time spent at each of those stages actually is a constant proportion at each temperature. They may grow faster at higher temperatures, but the proportion of time spent in each stage is a constant. That may be related to this idea of a master reaction model, or it may be helping us along. Also, metazoa have thermosensory neurons, which can trigger a hierarchical response right through the entire organism as a result of heat stress. So maybe that also has something to do with it. These are just ideas that may be connected. But the implication of these fits that we get, which, whether it's multicellular or unicellular, is that such growth is always limited by the thermodynamics of enzymes. But what is the nature of this rate-limiting reaction? Now, we will admit up front that not all of the parameter estimates are quite in line with what you might expect, but they're not implausible. It is still possible that the underlying mechanism may be more complicated than a single rate-limiting reaction. It may be, for example, possible that there's a metabolic network and this property comes out as an emergent property of that network. It just seems a bit improbable to me, since it has to do the same thing in all of these organisms. But if it is a single rate-limiting reaction, we can speculate on its nature. We know that protein denaturation is modified and controlled by chaperones and heat shock proteins. And some evidence for that is indicated by the importance of some chaperones becoming silent as they undergo sudden heat stress. So we're hypothesizing that there's a rate-limiting reaction which is linked to something to do with protein folding. And this process must have been conserved heavily during evolution, since it's present in eukarya bacteria and archaea. And if it has been conserved during evolution, it must have been present in the last common universal ancestor, or may have been. Either that or it's evolved more than once. Now, I think that's amazing that we can actually talk about insect growth rates and bacterial growth rates, and that leads us to say something about the origin of life. So we're dealing with a fundamental process, and speculatively it might be one of these topics. It might be natures, a trigger factor, an isomerase, something to do with protein disaggregation, or the maintenance of ATP availability. Some of the sorts of things it might be. So in conclusion, in the 2012 paper we used taxonomic groups, 3 taxonomic groups. We've shifted away to using thermal groups because that gives us a better model fit. We get results without using protein data, but we're predicting protein results. So we're bridging a gap between biochemistry and whole organism biology. We can describe how, it doesn't matter if you're a unicellular organism or a multicellular organism, we can predict how fast you'll grow under non-limiting conditions, and we can obtain estimates of thermodynamic parameters and even their relationships. So we've got a universal model which can fit, at least at the moment, to rescaled population growth data. We don't care if you're a prokaryote or a eukaryote, we don't care if you're a unicellular or a multicellular organism. All we care about is what your thermal group is, what your T-opt is, perhaps. And that suffices to predict your relative growth rate's response to temperature. Along the way, we've made some progressive advances in how we fit this model, and most recently we've been using adaptive direction sampling. But what's the bottom line is that it's remarkable that we can predict the growth rates of unicellular and multicellular organisms that have evolved over 3 billion years using one model. So in the future, we'll obviously go on to try to explain something about, or investigate something about, the nature of the denaturation curve, for example. We need to simplify the model. In order to apply it and embed it in other systems, it needs to be made more practical to apply. Once that's done, it can be applied more generally in things like climate change models. Perhaps we can model the ranges of test species as temperature changes. Perhaps we can predict tipping points in ecosystems as a result of temperature changes. This connection with LUCA is very intriguing, so that might lead to something. And there's a hot topic at the moment, which is the ancient proteins, so there may be a connection there as well. Personally, I think that if we can to predict what the thermal limits of life, the temperature limits of life are on this planet, or the maximum rate of growth at any particular temperature, that would be quite something. And that would certainly be of interest to astrobiologists. That's the end.